At the conclusion of the presentation of golf course construction, we were ready to establish turf grasses. At this point, we will look at how turf grasses are bred and a little about their genetics. Listed here are the three major means of how turf grasses reproduce. The first method we will discuss is Kentucky bluegrass. It is unique among the turf grasses because it reproduces apomictically. Apomixis is a reproductive mechanism that bypasses the sexual process, meiosis, and allows the plant to clone itself through seed. Apomixis is an ideal system of seed production. Once a superior genotype has been selected, a cultivar can be increased directly as seed. Apomixis provides a high level of uniformity and stability within a cultivar. The difficulty from a breeding view is apomixis competes with the ability to cross-hybridize. When we think of Kentucky bluegrass, we often associate it with the state of Kentucky. Although the pastures throughout the state were established at Kentucky bluegrass, it is not native. Kentucky bluegrass is native to Europe and Eurasia regions. This is Terceira, one of the islands that make up the Azores, which is part of Portugal. Kentucky bluegrass is widely used as a pasture grass for animal grazing. I wonder how long it took back in the day to make these stone fences. Even on this island in the Atlantic Ocean, traffic jams can occur. Since we are in Europe, I thought we would return to Ireland and Culzean Castle that we last saw under construction. The castle is still there. The tees, greens, and fairways were seeded to creeping bentgrass. The rough areas, however, are an assortment of grasses, including Kentucky bluegrass, which you can see in the brown area that's been mowed down. This photograph is from Carton House Golf Club, which is down the road from Culzean Castle. The point I want to make is that Kentucky bluegrass was established primarily as a forage grass seeded with common types. We ended up with a mismatch of bluegrass. This all changed when Joe Valentine, who was a superintendent at the Marion Golf Club in Pennsylvania, discovered a large patch of Kentucky bluegrass, a clone, that performed much better than the common types that made up most of the bluegrass on the course. After testing, Marion was released in 1947 by the United States Golf Association Green Section and the Agricultural Research Service, which is part of the United States Department of Agriculture, known by the abbreviation USDA. Marion was the first of the low-growing, dark green, disease-resistant, improved Kentucky bluegrasses. Its strength was in its low, compact growth habit, tolerance to low mowing, and resistance to leaf spot diseases. Since it was apomictic, it produced genetically pure seed. If you planted Kentucky bluegrass between 1955 and 1970, there was a good chance you planted Marion, especially on golf courses or other high-maintenance turf situations. Prior to the 1970s, other improved cultivars were ecotype selections like Marion. In other words, from a stand where common Kentucky bluegrass had been established, you might find a patch that looked different, maybe dark green, dense, or low-growing. You would then select that clone, establish it in a seed field, and then harvest the seed and sell it as a cultivar. In the late 1960s, Dr. Reed Funk at Rutgers University discovered in greenhouse studies that Kentucky bluegrass flowers open between 1 and 4 a.m. and shed pollen. This led to the development of a greenhouse hybridization technique for Kentucky bluegrass. This meant a new Kentucky bluegrass cultivar could be developed by hybridizations of two parents. No longer did breeders just select new ecotype clones from existing populations. Currently, there are over 200 cultivars of Kentucky bluegrass. I have listed a few as examples of ecotype selections where clones were found in different stands of Kentucky bluegrass and hybridization cultivars. In the ecotype column, I have listed Ken Blue, which is an example of a common type of Kentucky bluegrass cultivar. Its selection came from a Kentucky bluegrass pasture. The hybridization cultivars came from crosses of two parents. The asterisk represents the mother plant. For example, the cultivar Ginny. The mother plant was Julia and it was pollinated with limousine. Now you may notice that Ginny and Perfection have the same parents. When Kentucky Bluegrass parents are crossed, they can produce thousands of offspring or F1 generation plants. From this cross, at least in this case, two of the offspring were selected with different characteristics, which led to the cultivars Ginny and Perfection. Turfgrasses that produce seed, excluding Kentucky Bluegrass, are known as synthetics. In these hybridizations, a number of genotypes that have been selected for specific traits are crossed. In this diagram, an overview of the steps are outlined in developing a new cultivar or variety. Parent plants are selected, for the most part, from the field. The plants selected are based on genotypic characteristics. In other words, color. Maybe a parent is a darker green color, or is resistant to specific diseases or a disease, or has good shade tolerance, for example. The genotypes selected are propagated to increase the number of plants, and then they are allowed to cross-pollinate. And then when the seed is produced, the seed is harvested, and then it is planted to evaluate the number of factors, including seed yield. It's not a very effective cultivar if it can't produce seed. The plants produced from seed are evaluated in various environments. If this new cultivar is judged excellent based on the phenotypic characteristics in the field, the genotypes or parents are used to create the new cultivar and increased and established for seed production. In the United States, seed production fields for cool season turfgrasses are found in the Northwest. Oregon, for example, is the primary turfgrass seed producing state. For warm season turfgrasses like seeded Bermuda grass, the fields are primarily in Arizona, New Mexico, and maybe Southern California. Yuma, Arizona is a center of seeded Bermuda grass production. With synthetic cultivars, the number of parents used to produce a cultivar can vary greatly. In the case of creeping bentgrass, the number of parents in the table range from 3 to 203. This photograph was taken in a Pencross seed field in Oregon. Pencross has 3 parents, which have been established here, and now will be allowed to cross-pollinate. Once seed is produced, it's harvested and then bagged and sold as Pencross. In the following photographs, I'm going to make a few comments about seeded cultivars. This photograph was taken during seed harvest in Oregon. The mechanism of harvesting is similar to corn and soybean being harvested in the Midwest, similar types of combines. However, the combine header, the front part, which is the cutting platform, is different, setting much lower to the ground. This picture shows a creeping bentgrass green and perennial ryegrass fairway and rough. Phenotypically, the green and fairway are uniform looking. All the plants in each situation look the same. Although the creeping bentgrass green and perennial ryegrass fairway look uniform, each of the seeds that were used to establish a green or fairway are a little different genetically from each other. Synthetic cultivars are much more genetically diverse in comparison to Kentucky Bluegrass, where each seed of a cultivar is theoretically genetically identical. Relatively recently, improved varieties of seeded Bermuda grass have been developed for turf. Riviera, shown here, is a seeded variety resulting from intercrossing 3 parents with excellent turfgrass characteristics. This soccer field was established to Riviera Bermuda grass. The stadium is Mambubela Stadium, located in South Africa near Kruger National Park. Just a point of interest, the seats are patterned to represent the stripes of a zebra. The stadium was built for the 2010 FIFA World Cup games in South Africa. The 18 orange structures resemble giraffes. The fairway in this photograph was taken at the Royal Sydney Golf Club in Sydney, Australia. The seeded variety of Bermuda grass, known as Santa Ana, is widely used through Australia as a golf course fairway turf. The last breeding method to be described is vegetative. Basically, a clone is selected from a population, increased through propagation, and then tested in various environments. If the selection is to be sold as a variety, it is increased vegetatively, normally in a sod field situation, and then sold and established by sprigging, plugging, or sodding. Bermuda grass is the most common turf species that varieties are developed and vegetatively propagated. The Bermuda grass vegetative varieties come from the hybridization of Synonon dactylon and Synonon transvalensis. The hybridization results in sterile offspring. The chromosome number of Synonon dactylon is 36, and when crossed with Synonon transvalensis with a chromosome number of 18, results in a sterile hybrid of 27 chromosomes. In other words, the seed produced from the cross are sterile. From those offspring, however, maybe a few might exhibit outstanding turf qualities and be selected out and propagated to produce a variety. Tifway and Tifgreen Bermuda grass are a result of this cross. Other varieties or cultivars of Bermuda grass have been vegetatively selected from existing hybrid Bermuda grass as seen here. In some cases, irradiation of Tifgreen has been done to cause mutations, and then from these mutations, selections have been made. Just as a note, Synonon transvalensis origins are in Africa, around what used to be called the Transvaal region in northeast South Africa. The province was broken up into 4 provinces in 1994. Synonon transvalensis is a perennial that has very fine texture. One of its common names is African Bermuda grass. To finish this presentation off, this is a Tifgreen Bermuda grass screen in Australia.